Regulation of RNA stability. Alternations in RNA stability may take a considerable contribution to gene regulation in eukaryotes. Regulation of RNA stability may operate in either nucleus or the cytoplasm. Now, regulation at nucleus. Some genes are known to produce heterogeneous nuclear mRNA, that is hnmRNA, which in some cells is never processed to mRNA and never transported to the cytoplasm. These transcripts are degraded in the nucleus before they can be processed. Now, regulation at cytoplasm. The stability of mRNA can control the availability of the mRNA for translation in cytoplasm. The stability of mRNA of different genes may vary considerably in their half-life. Some genes that codes for abundant product with a long duration of action, such as the beta hemoglobin gene, may produce mRNA that has a long half-life. Other genes that codes for a product with a short duration of action, such as those of growth factors, may produce mRNA that has a short half-life. Many growth factor mRNA have half-lives of less than 1 hour. Sequences within the 3' untranslated region of unstable mRNA that are responsible for their rapid degradation have been identified. There is also strong correlation between length of poly A tail and the half-life of the mRNA. The longer the poly A tail, the longer the half-life of the mRNA. Now, let us see some examples of gene regulation by RNA instability. The stability of alpha and beta tubulin mRNA may change depending on the availability of alpha-beta dimers. The more alpha-beta dimers are available, the less stable are alpha and beta tubulin mRNA. If tubulin polymerization is inhibited with colchicines, the amount of alpha-beta dimers increases. Further, alpha-beta tubulin dimer production is stopped through selective destabilization of alpha and beta tubulin mRNAs. Now, inhibitors of protein synthesis. Different types of compounds inhibit protein synthesis, such as therapeutic drugs, toxins and plant products. Let us discuss these molecules one by one. First one, therapeutic drugs. A number of antibiotics that inhibit protein synthesis have proven to be effective antibacterial drugs. Some examples are A. Streptomycin. It binds to 30th subunit of bacterial ribosomes. It causes misreading of mRNA and at high concentration. They completely inhibit initiation complex formation. B. Tetracycline. It binds to the 30th ribosomal subunit and inhibits the binding of amino-acyl tRNAs. C. Chloramphenicol. It binds to the 50th ribosomal subunit and blocks the peptidyl transferase reaction. D. Erythromycin. It is an antibacterial drug that inhibits the translocation reaction by binding to the 50S ribosomal subunit. E. Rifamycin. It blocks initiation transcription by binding to RNA polymerase, which prevents RNA synthesis. Second, inhibitors. Other antibiotics inhibit protein synthesis and are used for research purpose but have not proven to be good therapeutic drug. These include A. Cyclohexamide. It inhibits protein synthesis in eukaryotes by binding to the 60S ribosomal subunit and inhibiting the peptidyl transferase reaction. B. Puromycin. It is an analog of the amino-acyl adenosine moiety of amino-acyl tRNA and leads to premature termination of translation in both prokaryotes and eukaryotes. It competes with amino-acyl tRNA for binding to the A site. In the A site, peptidyl transferase links its amino group to the carboxy group of the peptidyl tRNA in the P site. This linkage causes premature termination. Third one, toxins. The mechanism of action of several potent toxins is through the inhibition of protein synthesis. A. Diphtheria toxin. It is produced by a lysogenic bacteriophage that infects Corynebacterium diphtheriae. Diphtheria toxin causes inactivation of EF2 by attachment of ADP to EF2, and inhibits translocation step of elongation. Because diphtheria toxin is catalytic, only small amounts are needed to be toxic to the cell. B. Resin. It is a protein produced by castor beans. The resin toxin is N-glycosidase and removes a single adenine from 28S rRNA. This depurination inhibits protein synthesis in eukaryotes. C. Ribosomal RNA specific nucleases. Other toxins inhibit protein synthesis by the specific cleavage of rRNA. Alpha-sarcine is a toxin produced by the fungus Aspergillus giganteus, which cleaves 28S rRNA. This specific cleavage leads to the inhibition of aminoacyl tRNA binding. Colicin E3 is another example of ribosomal RNA specific nuclease. It is secreted by some strains of E. coli, inhibits protein synthesis in other bacteria. It does this by specifically cleaving 16S rRNA, which leads to the inhibition of initiation.